thank you very much. Am I screaming too much? Because I tend to do that too. My name, as you know, is Montserrat, or Montse. So I really thank all of you for being here. It's actually my pleasure to be able to share some of the things I've done and like all the things I absolutely love with you. And hopefully by the end of this talk, I accomplished two things. One is that you think chemistry is actually kind of fun. And the second one is that you learn something. So hopefully those goals are accomplished. So the title of my talk is The Research at the Frontier of Chemistry and Biology. So as Terry said, I'm a chemist, so a chemist by training, but I absolutely love biology. So really, I'm really interested in understanding biology from the chemical point of view. But so first, so what is it your first thought? And be honest with yourself. What is your first thought when you think about a chemist? Wow. Or a chemistry?
Awesome.
OK, he may be honest because he's a chemical engineer, but the rest of you. So is this the first thought that comes to your mind? Like, you know, we all these crazy people, you know, like the classical, like they mix two things, and then like your face is all. So, but no, really, that's not quite who we are. But maybe your first thought was this. Do you know what that is? An awesome show, but that's not who we are either. So, and FYI, the experiments they do in that show are way more dangerous than, you know, what it looks like. So never do that. So, but now, really, what do we do as chemists? And let me give you a few examples of some of the chemists that are really awesome, and they have done like all these amazing things. So first we have Arthur Handen and Euler Chaplin, and sorry, I cannot pronounce them. But what they did is that they actually studied the fermentation of sugars and enzymes. And we all know what that means, right? But really, the discovery of all this has led to a lot more things and the understanding of a lot of different things, like how molecules work, how they look like. So, you know, they really contributed a lot with doing some fun things. So another one, and we all have heard about this also, the Curie couple, right? So they were the first ones to actually synthesize radioactive materials. And first of all, I mean, these are amazing chemists because they were actually making radioactive materials without any protection whatsoever. They didn't quite understand what was happening. Well, until later they realized what was happening. But at the time when they did all these experiments, there was no protection against radioactivity. And also, so you know, Marie Curie, she's the only person that has won 2 Nobel Prizes, one in chemistry and one in physics. So that's what we have to look for. OK. So, but really, without them, there wouldn't be such thing right now as nuclear medicine. So we all have heard about that, right? So what do they do is that they put in these minute amounts of radioactive material in you so they can figure out, OK, maybe there's a tumor here. Or, you know, in some other cases, it's like they can target cancers using these radioactive materials. So the discovery is really, really important, and it has allowed the understanding of many, many different things. So now these three, Boyer, Walker, and Coe. So this is a little bit more recent. This is in 2008. So what they did is that they elucidated the enzymatic mechanism for the synthesis of ATP. OK, who knows what I'm talking about here? OK, Brittany, thank you very much. Yeah, the pros, you know, those biochemists. But really, this is important. Why? ATP is the cash in the money. So whoever understands where money is, we can understand a lot of things, right? So what they actually did is that they understood how money is made. So it's really important, and it has led to a lot of understanding of many different things. And not only that. So those of you that are engineers, I'm going to challenge you to develop. This is an actual, it's a protein, but it's like a machine. It's like a motor that moves like this and is producing enzymes or certain molecule that is ATP that is basically money. But it's beautiful. I mean, it's like an engine, okay? And this was done first. And it's super efficient, so it's really awesome. And again, of course, one of the classes I taught, it was molecular cell biology. And every time I teach this, it's like, oh, this is my favorite protein. Although I say that like every other class, but that's okay. Okay, so finally, one other one that is really important is, and this was the Nobel Prize in 2009, is Ramakrishnan, States, and Jonath. What they did is that they understood and studied the structure of the ribosome. So who knows what ribosomes are? Okay, a little bit more, so I guess they are more famous. So pretty much what they are is the machines in your body that are going to make proteins. We all know about proteins, right? So that's why it was really important. So this is a picture of ribosomes. So really, a lot of chemists have devoted their lives to understand all these biological processes from a chemical point of view. So let's define chemistry a little bit first. So chemistry is the science of matter, and especially the chemical reaction, but it also talks about composition and property. So also what we are trying to understand is interactions with other atoms and specifically the properties of different chemical bonds. And all of you that are in my organic class, we've talked a lot about chemical bonds and why they're important. So these are all the elements here, and then if you cannot see this, you can look at the periodic table over there. So there's a lot of them. But really, what we focus more in is just a few of them, which are carbon, hydrogen, nitrogen, oxygen, and sulfur sometimes. So, but what is biology? So biology is a natural science that is concerned with the study of life and living organisms. So it's going to include their structure, function, origin, evolution, distribution, and taxonomy. So really, kind of, as I told you previously, what I'm really, really interested in is to understand the living organisms and how they work, both from a chemical point of view. So you can think about it like, well, I really like cars, but I don't care so much about the car itself. I want to see how the engine works, or how like the steer is made, or things like that. So I want to look at things in a little bit more detail so I can understand them. So that's what I'm really, really interested in. So what I want to see is life from a chemical point of view. So that's the general focus of my research. And I want to talk to you a few of the research I've done here, and then some others I'll just like mention the topics. But before I start, I really, really want to thank the people that made this possible. And the people that made this possible are all the students that have worked with me. I mean, my students, they work really, really hard, and I hope they love what they are doing, but I think I can see it on their faces. But that's the first thing I want to do before I talk to you about my projects. So for the development of novel biomaterials, I had 4 main students working with me. Desiree, that graduated, and she's in Purdue right now, doing her PhD. Tyler, he graduated, and he's doing his master's in biomedical science, policy, and advocacy. Elizabeth Santos, she's in MSU right now doing her PhD. And David Lecronier, which is in medical school right now in MSU. So those are the students that work with me, and my collaborators are Xinguo Chen in the Southwest Research Institute, and Pat Atkinson here in the mechanical engineering. So thanks to them. Then for the other project, I'm going to talk about the study of antioxidants. Again, Desiree, Elizabeth, and Randall, that he just graduated. He's working at Pfizer right now, and Brittany. Okay. And thanks again to my collaborator, Liwa Wang, which is a faculty here. So the other one I will talk about is the novel of synthesis antibacterial. So my collaborator in Michigan State University, Professor Borgen, and Elizabeth is the one that did most of the synthesis here. So the biosand filters, so these are members for EWB, and this is the ones that were to the last trip. Later I will show you all the names for a lot of people that actually work really hard on this. So David, Justin, China, you should be here. Maybe he's late. Oh, there, sorry. Nikki. So Nordine, he's not here right now. Nadine, thanks. Elizabeth again. And many thanks to Laura Sullivan. She couldn't be there with us, but she was really a very important support. So these are the projects I will not be able to talk to you about, but it's the development of a novel method to study proteins. So she's Lacey. She's my first undergraduate student. So like right now, she's working in GM. Simona, she's in the U of N Dearborn, and Professor Borgen again. Then the novel green production of phenol and enzymatic reaction at high temperatures. Sam, Mariam, I saw you over here. Patrick, he's on the other section. So luckily we have this Patrick that is from this section, so we can call one Patrick on. And then Jonathan, thank you, and Ali. And then finally the coding of polymers, which is Nikki and Justin, again. And then Susan, over there, thanks, Mary, and Ali. So these three I will not be able to talk to you about, but hopefully they can tell us later about those projects. So, and again, feel free to stop me, interrupt me, or whatever if you have any questions, okay? So the first project I wanted to talk to you about is the development of new biomaterials. So we all know how, materials are very important. But specifically in the orthopedic arena, developing new materials is extremely important because it's a market that is growing significantly. So about 1.8 million of artificial joints happened in 1988, and about 4.9 million of fixation devices. So I'll talk to you about the difference of those two. But here what we are looking at is the trend of hip replacements in 2006, I think. Oh, sorry. Over a series of years. So we can see all the way to 2004. And here, the total knee replacements. So what you can see is that there is definitely an increase in the number of people that needs total knee replacement or hip replacement. So these are very important markets. So what are these made of? So, well, let's look at all the implants, first of all. So they will be made of metals and polymers. So we have implants that are facial prostheses. We have dental. We have vascular grafts. So it's pretty much what they put in your veins whenever you have a procedure. We have hip implants. We have knee implants. And then we also have legs and arms. So in general, usually, these 1, 2, 3, 4 are going to be put in people when they are older. So the market for this 1, 2, 3, 4 tends to be an older population. But on the other hand, these and these leg and arms tend to be also market in the young population because they mainly occur from sports accidents or car crashes. So the nature of how and what you do with these implants is different for young people and old people. So I am actually interested on trying to improve both arenas. One, implants for younger people, and implants that are going to be mainly for older people. So really, the polymers, they have shown to have excellent biocompatibility. But who knows what that means? So really what it means is that they put them in you, and they don't kill you. So that's really what it means. So you can tolerate them pretty good. So the only problem with them, however, is that over time, they can erode a little bit, you know? It's like kind of your shoes. You start walking, and they start, you know, all these little particles come off slowly. But, you know, you walk everywhere, so who cares if there's like a mini particle just, you know, here, and then you go to your house, a little bit over there, a little bit everywhere. So that's not a big deal. But what happens when you go home, and then you only walk in your home, and just imagine doing that over and over and over, it actually accumulates. So that's the same thing that happens in the body. So all these little particles accumulate. And then part of the problem is that your body can identify them. And it's like, oh, that's kind of bad. You know, after a while, your body recognizes them. I don't want that here. So it starts attacking them. And that's a problem, because then you cause this disease that is called periprosthetic osteolysis, which is that the bones around the implant start dying. And why they start dying? Because you have these mini particles next to your bone, so your cells, that some of them are like police, they are checking that everything is okay. So they come here, oh, it's not okay, it's not okay. Oh. So they destroy this one, but when they destroy this one, they bring some of the bone with it. So that's bad. So that can be a problem. And the longer you have the implant, the more likely is that problem to occur. So here, this is pretty much what we are seeing is either an infection or the implant is being rejected. So by one of the reasons is erosion of the microparticles. So here you can see that the entire tissue is damaged. So how do you solve that problem? So, well, this is one of the projects I will not be able to talk to you in detail, but you can actually modify the surface of those materials so you can trick the body. And so you can actually have your implant here, and like the little particle comes off. And then you can trick the body to tell, OK, this particle looks just like you, like your body. So don't kill it. Just leave it there. It's OK. So that's one of the things we are trying to do by modifying the surface. But then another thing that we are trying to do is that we can modify the surface so there's actually less emission of those particles, so less corrosion or less wear. So we are trying to accomplish those two things. But we don't have time today to talk about that. But so how about the other ones, the long-term implants, like these ones, that belong to part of the population that can be young? So these are mainly made of stainless steel, an alloy of chromium and cobalt, titanium. And there are a few ceramic and polymers. But they are actually very good because they are inert. You know, you can have them in yourself for a long time. They have the mechanical strength that is needed for you to be able to walk and do whatever else that you need to do. But some of the problems, especially in long term, is that they are going to corrode. And when they corrode, they are going to have that problem, the immune response. Then, well, I'll talk about that later. But the density and the mechanical strength is very different from the bone. So you are having all these extra problems about growing your bone more in one places than others. But you know, we can talk about that later when we are out of here. But another big problem here is the secondary surgery. So in a lot of the cases, a secondary surgery is going to be needed. So how do you solve all these problems, or at least some of them? So you develop an implant that once you put it in, is going to do two things. One is going to help the bone to regrow. And then the other one is that it's going to disintegrate. It's going to disappear. It's going to biodegrade. So that was our goal in mind. So we wanted to develop an implant that you can put in, and then it's going to aid bone regrowth, and it's also going to biodegrade. So that was our goal. And then for that, we chose this material that is called magnesium. So I know all of you that are into cars, you've heard about magnesium, right? Like all these rims and stuff, like pretty cool. So why is magnesium so cool? For making, exactly, it's very light, right? So what is good about magnesium is that when you look at the properties of magnesium and the bone, they are very similar, both in density and strength. So that's a plus. So then also it's biocompatible. Why? And this is where chemistry comes in. So we have magnesium, okay? So magnesium, if you put it in water, which is not a very good idea sometimes, but let's just say, you take magnesium in water. So what you're going to form is mainly this magnesium hydroxide, which you should know what this is. I bet you have this at home. What is this? Exactly, yeah, all these antacids, not all of them, but some of them have this. So you know this is not going to be that bad for you, right? And then you do form gas. This is, I'll talk about this in a minute. But then also in the presence of chlorine ions in your body, then you can form this magnesium chloride, which is very abundant in your body, so it's okay. So when you actually look at the amount of magnesium, notice here I have ion, so, but we'll talk about it later. This is, we have in an adult, we have actually 22 grams of magnesium, so it's a lot. So really, having toxicity from magnesium is really, really rare. You really need to either have a condition or take a lot of it. So it's actually pretty decent. The toxicity is relatively low. But again, the problems. Here, we form hydrogen. So that's a bad thing. You have gas. So if any of you, well, we all have gas in our stomach, and we know how uncomfortable that is. But can you imagine to actually have gas in your muscles? That's actually pretty uncomfortable. And if you put large amounts of it, then it can be bad. So that's a big problem. And then also, it degrades very rapidly. So you know that you can take, magnesium and make car parts out of it. But the catch is that the magnesium is not pure magnesium. It's an alloy. So you add other metals to it. So one of the metals that you can add to is aluminum, zinc, and so on. But those are toxic. So then for our studies, we choose one that had very small amount of magnesium in. Sorry, very small amount of aluminum in, which can be toxic. So we chose that alloy here. So the experiments I'm going to be showing you are with magnesium that has a little bit of aluminum in it, which could be a problem. But OK, so what did we have to do? So the first thing is that we had to somehow cover the magnesium so it will degrade slower. So that would allow for the production of gas, hydrogen gas, to be slowed down. So then the body can deal with it. So that was our first goal. And then once we coated the samples, we needed to test them to see whether they will stop corrosion or not. So we did tons of samples. So I'm going to show you only 3 of them just to simplify things. So what we have here is pure magnesium. And then here is 3 different coatings. So what we were measuring is how much magnesium is going to be released. Okay? So the more is released, it's going to be the higher this lane is going to be. So you can see that these 3 basically were better than without the coating. Okay? So we released less magnesium here than here. So that was good, promising. So now we want to do some toxicology studies, right? What if we did something awesome and we coated it pretty good, but then it's going to kill you? So you have to go, when you're doing all these experiments, you have to go slowly. and you have to think about the money you're spending. So the cheapest way of testing this is toxicology studies in vitro. So what this means is you're just doing a test tube. So we took some cells, and then we grow them in the presence of these implants. So what we did 2 different types of tests, but the bottom line is that it was actually OK. It was better than magnesium. So because of these toxicology studies were encouraging, then we move on, and then we move to studies in vivo. So now what we did here is that we actually took, and this, if you are grossed out easily, don't listen. But so what we did is that we took rats, and then this is the leg of the rat. So, well, I'm short, so here, you can see here. So this is the leg, OK? So then right here, well, you can see it better here. So right here, we put the implant. So right next to this bone here. OK, here you can see that's the implant. OK? So these pictures are shown at the end of the study. So what we were looking for is we put in the implant, and then we were monitoring how safe or how good those animals were doing. And so we tested some protein and things, and I'm not going to bore you with all that. But basically, what we found out from this study is that the animals that did not have any implant were exactly the same as the ones that had the implant. So that was good news, right? Because that means there was really no difference. The animal didn't see any impact. And then another thing that was very encouraging is when we took the coated implants out, we could see this. So these are 2 different views. And what you can see is that it actually remodels. So what this is telling us, in a few words, is that the body liked it. So it was actually taking things out and putting other things in. And not only that, when you actually, so the top one, it's a picture of an implant that did not have any coating on. Then this is a picture of a bone. This is a picture of our coated implant after we took it out. So you can see that these 2 are actually similar, OK? Like the morphology. And then we did some studies trying to see which atoms were in there, and they were similar also. Not exactly the same, but they were similar. And what is very encouraging is that you can see that if you have very similar morphologies, then the cells that are going to make bone grow, they will like this morphology. So that was also very encouraging. So then once we figure out the implants were not toxic for the animals, and that we may have the ability of regrow bone, then we went ahead and we tested for that, bone regrowth. So how do you do that? Again, this is kind of cruel. What we did is that now we went to rabbits, because you cannot do this in rats. So you take the bone and you cut it. So you cut it to a size that is called the critical size. And what that means is that we know experimentally that when the bones are cut really far away from each other, they are not going to grow back. Okay? So we knew that. So what we did is that we cut the bone in that size, and then we put in our implant. So just to summarize everything, pretty much this is showing that. So this is how we cut the bone here and here, OK? And then we put in the implant. So then we monitor this by X-ray over 12 weeks. So these are different weeks. And then what you can see is how bone actually starts growing around this. Okay? And these are X-rays. And then we took a CT scan of the animal once it was dead, once we put it out. So it's basically this. So this is, if you look at the bone and then you take a picture this way. So what you can see here is all this is new bone. So there's no other material so far that they've shown this. And this is still our implant. But you can actually see how this implant is not the way it was before. It's been degraded a little bit. And then you can also see, like from the top, this is the new bone that is forming here. And then this is the alloy that we had. So all these results were really promising. There are still some issues that we have to deal with just to make this better. And unfortunately, we kind of run out of money, so we have to stop for a while. So, but this is really promising, and we were able to develop a biomaterial that will degrade over time, and it will allow bone regrowth. So that was pretty exciting. So if any of you is really interested in that, we have a publication, so I can give you the reference if you want more details.
Can I ask you a question?
Yes.
I mean, is there any difference in degradation after the bone forms around it? Is it still degrading at the same rate, or is it less, or do you know?
We are not 100% sure, but it remodels. Because actually, bone is made of magnesium also. So it takes some of the magnesium in there and deposit it. So it's a little hard to exactly pinpoint that down. Any other question? OK. So now the other project I want to talk to you is cancer antioxidants and the synthesis of antibiotics. So this is something we, have you ever thought about how you make drugs? And like if you want to be a millionaire, you can actually get into business of making a very successful drug like Lipitor, which we sell about 12.9 billion per year. So it's like a huge market, right? And we all know at least somebody that is taking Lipitor, right? What is Lipitor? Exactly, an anti-cholesterol drug. But did it ever occur to you, how do they come up with these things, right? So it's actually a very interesting question. So the first thing that you have to identify is the problem, excess cholesterol. So then the second thing is that you have to figure out how cholesterol is made. And then we find out that actually most of it, we make it ourselves. We don't eat it, so some of us are cursed, some others are not. And then, so it's transported in the blood by these two different cells, the HLD and the LDL. So this is basically what they measure when they do your blood tests. So this is how they measure how much cholesterol you have. But so basically, if you have a lot of it, it's going to lead to arteriosclerosis, which closes your veins, and then you get like cardiovascular problems. So this is the structure of Lipitor, which we call the atrovastin calcium. The Lipitor is the trademark word. So, well, how do you solve this problem? Well, we figure out the synthesis, and I'm not going to show you the entire thing, just a little piece of it. But what is important is this particular chemical, which is called melavonic acid. This is the precursor of cholesterol. So you can think of it, this is the one that is the bricks if you want to make a house, right? So if you don't have bricks, you don't have a house, right? So that's pretty much the idea. How about if you stop this from making? So then you don't have that. So what you have to identify is, again, what is making that? So I know for most of you, this is just like a crazy drawing. But what this is just a picture of a protein, OK? So this is the guy that makes this one, sorry, the melavonic acid. So what this is showing is just a picture of the factory. So this is the machine, and this is what you make, like this part here. So you actually can block that, right? Just literally like machines. So you can put something in there to stop it from working. So that's what Lipitor does. And basically, like this structure here is very similar to this. So let's think about it this way. So what you have is the water running, right? So what happens in the people with high cholesterol is that that water runs too much, and then you have a flood. Sucks. So sometimes you can actually change that by putting the correct washer. right? And then you just fix the problem and fine. But sometimes your valve is so messed up that it doesn't matter how many times you put in the washer, it's still not working. So you have to be drastic. And you just have to plug it in and just seal it, you know. I'm very bad for that, so I have to call somebody. But that's basically what you do. You just block it. So it's the same idea. Lipitor, and again, for those of you that know exactly what I'm talking about, I'm making a lot of assumptions, so please bear with me. But basically, you block this protein from working. So then it won't make cholesterol. So that's exactly how drugs work. So it's very important, first of all, to understand the protein that you want to target. And then another important thing is like, OK, I identify my drug, which is this, which is Lipitor. But now, how do I make it? I cannot just go and like buy it from the market, right? You have to make these molecules. And that's where the big question and problem is. So in very general, and I know I'm an organic chemist, so I love this, but I'm not going to show you the rest. So we can just separate this into two main parts. So just imagine you take your car and then you separate the engine from the rest. So to make the engine, you're going to need 6 steps. So you need to go through 6 different processes to just make this one. And then you have to go through 2 different steps to make this one. Then you combine them, and then you get that one. Okay? So you actually have to learn how to make these things and the most efficient way. So what I'm interested in is cancer. So we all have heard about cancer. We know how bad it is. So since I'm running out of time, I'm going to jump on this. So the causes are chemical carcinogens, environmental carcinogen, viral, and radiation. And so some of the ways that you can prevent this is with outside agents, so antioxidants. And the body has a natural way of dealing with bad cells, which is apoptosis. So I had a picture here, but we are running kind of short on time, so I'll show it later. But apoptosis is a very important process, and it's basically cell death. Okay? So the cell kills itself if it's bad. So it's very good and important because there are certain proteins that are going to be key for this. So some of this is basically like, you know, you have a gang. So the people that is going to take care of it is the police, right? So they'll make sure that they stay all either in their neighborhood or they go to jail. So it's the same idea. So some of these proteins are going to be very important on keeping track of cells. And if they're bad, it will trigger to die. So one of those proteins is this cytochrome C. So we are very interested on understanding how this cytochrome C works. So it's, again, involved in apoptosis and also in the respiration process. So that's one part of it. The other part is that we are really interested in antioxidants. So we all know that drinking tea is good for you, right? But you know why? Because of the antioxidants. So that's why now coffee is on the good side. Now coffee is not that bad anymore because it has a lot of antioxidants. But when you look at tea, especially green tea, has very high amounts of these molecules here that are called catechins. So we were really interested in understanding to see, first of all, if there is any relationship between these molecules here that are called catechins and cytochrome C. So what we found is that, yes, there is a relationship. And then we wanted to understand more about, well, what is that relationship? So maybe we could develop a drug later on you know, that involves these characteristics. So basically what we did is that we took molecules that were very similar to catechins. And just believe me on that. But you can see the pictures, how they all look kind of similar. But they had some differences. So we could test which parts are good and which parts are bad. So what we eventually concluded is that this is one of the catechins, okay, the simplest one. But we realized that this part is really important to have an interaction with cytochrome C. So what this means is that if we wanted to have a successful drug, we have to keep this, okay? So then, and basically we realize how it reacts with this protein here. And then, since I'm running out of time, so this is a synthesis of naprodicin. And so this is how much we have made. So you can see here, this is, we prepare this part of the molecule. We need this part. And then, so finally, I'll just talk to you very quickly about biosand filters. We took these biosand filters to Mexico. So we did it in basically 5 trips. So this is a pre-assessment. Then this group implemented some of them. And then this group implemented 45. And then here, this group, which is the one I showed the picture of, we analyzed them. So just let me tell you the results. So it's, since I'm running out of time. So we went back and we checked. And only 25% were working, 62 were not working, and 13% we could not find the people. So this may look like, you know, kind of disappointing to you. After we put 45, only 25% is working. But actually, it's really, really good rate, because usually when you put this on, there is, in 1 year, only about 15 to 10% of the people still use them. So that's pretty good. And then from the ones that were working, we tested some of them to see how much bacteria it removes, because basically that's what biosand filters are going to do. They will remove bacteria from water, and it's very cheaply made. So then we found that most of them, this is the bacterial removal. So they are all at least higher than 50%, except one of them. But it still removes some. So it's still better than what they will drink otherwise. And then we found out that because of the ones that were not working, it wasn't actually that bad, because 32% broke. We found this problem with our design. Then the other one, there was this very bad drought. So the water they were using was full of algae and bacteria. So basically, when they used it, it messed up the system. So that's what happened. So it's actually, it was a very nice experience, and you can ask them. I'm very proud of all of them. And we learned a lot, and it was a pretty fun experience. But unfortunately, we had to cancel this project due to the violence over there. So we were not able to go back. So thank you all, and I hope chemistry is fun. It's not that bad.